Hello, I'm Dr. Charles Knight, and this is my 10 Lectures on Evolution series that I'm recording for my Bio 414 Evolution class at Cal Poly here in San Luis Obispo. I work in the Bailey College of Science and Mathematics and in the Department of Biological Sciences. There's 7 other talks that I've recorded in this series, so check them out. But otherwise, please enjoy this one. This paper is on adaptive tail length evolution in deer mice and how it's associated with differential HOX-D13 expression in early development. It was published in Nature, Ecology and Evolution in April of 2024 by some prominent scientists, including Evan Kingsley and Hopi Hoekstra, who I'll talk about here in just a minute. But before we get into that, I thought I'd start with some fun videos that were part of the supplementary material in this paper. This is a forest mouse that's running across a wire, I guess you could say. Well, I'll just go back and we can watch. Well, I can't go backwards, but that was the forest mouse, and this is the prairie mouse trying to do the same obstacle course. Point here is that the forest mouse made it across this rod very easily. We can assume that the tail helped a lot in its stabilization, but the prairie mouse is like, nah, I'm good, can't make it. So I think it's useful to also think early on whether you think that the deer mouse has recently evolved longer tails or shorter tails. Keep that in mind as we go, and maybe we'll come to a kind of conclusion on that later at the end. But I'd first like to talk about the investigators here, Evan Kingsley, who's the lead author, and Hopi Hoekstra, who's the last author. I'd like to just say a few words about them. I apologize to the intermediate authors because in this class we don't have time to go into detail on everyone, but I encourage my students to do a little scientific vignette or a scientist vignette so we get to know these people who did this work just a little bit. I don't know Evan, but I'm really impressed with this paper and the body of work that it represents. I know that Evan just graduated with a PhD from Harvard with Hopi Hoekstra and now works at Rhodes College in Memphis, Tennessee. Before that, he had worked on voice box muscles. Whoops, I see that I misspelled box there. And he's had a great career so far, and I look forward to continuing to track what he's doing. But then, of course, Hopi Hoekstra is one of the most prominent evolutionary biologists in the field and is very famous for the pocket mice in Arizona that she studied, I think, as a postdoc after her work at the University of Washington on the left-hand side on grizzly bears. She shifted into a slightly more tractable system with the pocket mice and then has continued on with this work on mice as investigator at Harvard across the last 10, 20 years. Well, I don't know exactly how long Hopi's been there, but I do know that she's recently become the Dean of Arts and Sciences at Harvard. So I hope she's able to continue producing papers like this. I've always enjoyed reading Hopi's papers. So with that, I'll get started here with my take-home points from this paper. I always like to have like 4 take-home things for my students, like things that you can really just take to the bank. I always start with a really general one, that tail length variation has recently evolved between prairie and forest deer mice in the eastern US. This has been pretty obvious for a long time. This wasn't new to this paper. Been a long series of observations since the 1910s, 20s, showing that these forest mice have longer tails. And it's clear from this paper and other work that tail length has a performance advantage in balancing tests, so longer tails are better for getting across twigs or in this kind of test that they did. In this paper, they really delve into the genetics and do a QTL analysis with RNA sequencing and a gene phenotype public information database to really narrow down to what genes are causing this. And they kind of highlight HOX-D13 as a gene with major effects, a major effect on tail length. And they did some confirmatory experiments with CRISPR when they found that there was a 3 base pair insertion. They used CRISPR to insert that extra base pair, or sorry, that extra amino acid in the lab mice and looked at the effect on tail length, and it had no effect. I'm just spilling the beans early on. But so there was a negative result there, but I really appreciate that they reported that negative result, and I always try to emphasize to my students how important it is to report those negative results. Otherwise, we think that CRISPR works every time. But I was, in a way, happy to see an example where CRISPR didn't work. Not happy, but it just is reality, and we often don't see that reality. So kudos to you for reporting that. And then in the end, we kind of have to assume that there's cis-regulatory effects on HOXD13, since it doesn't appear perhaps to be the protein itself that is changed, although it does have one additional amino acid in the forest mice. But in addition to that, certainly regulatory changes may have the effect on tail embryogenesis or tail morphogenesis. So there's still more work to be done here. Probably years of work to continue Evan in his new position at Rhodes College. As I always do, I'm going to start with reading the abstract. Variation in the size and number of axial segments underlies much of the diversity in animal body plans. Here we investigate the evolutionary, genetic, and developmental mechanisms driving tail length differences between forest and prairie ecotypes of deer mice. We first show that long-tailed forest mice perform better in an arboreal locomotion assay, consistent with tails being important for balancing during climbing. We then identify 6 genomic regions that contribute to differences in tail length, 3 of which associate with caudal vertebra length, and the other 3 with vertebra number. For all 6 loci, the forest allele increased tail length, indicative of the cumulative effect of natural selection. 2 of the 3 genomic regions associated with variation in vertebra number contain Hox gene clusters. Of those, we find an allele-specific decrease in HOXD13 expression in embryonic tail buds of long-tailed forest mice, consistent with its role in axial elongation. Additionally, we find that forest embryos have more presomictic mesoderm than prairie embryos, and that this correlates with an increase in the number of neuromesodermal progenitors, which are modulated by HOXD13 paralogs. Together, these results suggest a role for HOXD13 in the development of natural variation and in adaptive morphology on a microevolutionary timescale. There's lots of words that I'm going to go over in the next slide in this abstract that I think are important to highlight. Caudal vertebra, the forest allele, Hox gene clusters, embryonic tail bud, and you'll see what I do on the next slide with what I call glossary notes. So the caudal vertebrae are the bones that make up the tail. And the forest allele, in this paper, it's the QTL genotype present in a forest mouse. They found 6 QTLs, each has a forest allele. And eventually we'll see down to the gene level, there's even a forest allele of the HOXD13. However, the genes themselves don't vary much, but we'll see how that goes here. Hox gene clusters, just important to know that there's 4 clusters in mammals, and humans have 4 clusters. They're labeled A, B, C, and D, and each cluster has approximately 13 genes, and we'll see this in a slide coming up. The embryonic tail bud is the mass of undifferentiated mesenchymal cells at the caudal vertebra tip. And we'll see this again in figures that follow as we try to understand how tails develop. The presomictic mesoderm is the before somite tissue. A somite is an individual vertebrae, essentially, that becomes the tail. These are presomictic. This is before they become somites. And the amount of presomictic tissue turns out to be important, and HOXD13 is playing a role in forming that tissue. The neomesodermal progenitor cells, or NMPs, are the stem cells for tail development, and they produce the presomictic mesoderm, and they're certainly where HOXD13 is operating. And the paralogs, just remember that paralogs are gene copies in the same organism, and microevolution is a term that's sometimes used for relatively short timescale evolution or smaller magnitude evolutionary changes. Remember that Sean Carroll suggests that macroevolution is microevolution writ large, and I tell my students there's really not that much difference. It's just the magnitude or timescale. All right, so I always have some textbook tie-ins to our textbook, which is the Freeman and Heron text that my students use. And this is figure 19.4 on the Hox genes. And we've seen this before. We're reading Endless Forms Most Beautiful, and that book's basically all about Hox genes, or a lot about them. This figure is just really nice. It's colorful. It shows the 4 cassettes, or four groups of Hox genes in vertebrates, and this is a mouse, for example. There's A, B, C, and D. And you can see how some of those cassettes, like the C cassette is missing Hox 1, 2, and 3, and 7, and I guess it's just those that cassette C is missing, but approximately 10 genes, but really drawing from 13 possible genes in each one of those cassettes, compared to the fly, which has reduced, well, the same genes, but a different different complement. Also important to remember that the Hox genes are expressed anterior to posterior from the tip to the tail in a predicted order, and you see the tail vertebra in that mouse embryo on the top right below my picture here, just right down there is they're the gray Hox genes, and the very last one is Hox D13, which is at the tip of the tail, and it turns out that this gene is important for controlling or contributing to tail length variation. And I got this figure from a different paper on neomesodermal progenitor cells, or NMPs, and they're like the Hox genes of, the stem cells of the developing tail. You see the NMPs become NMP spheres, which could then become neurons or skeletal muscles or cartilage, and really they're developing into those cells in the tail. Interesting to note that Hox D13 isn't shown on this slide, but TBXT is, and there's a tie-in paper that I'll talk about next week, maybe I'll post it here too, about how alternate splicing in TBXT may be partly controlling why humans don't have tails and why the great apes don't have tails, because there's a deletion of an exon in TBXT, and that perhaps leads to us not having tails. I'll talk about that paper next week. But let's just talk about tail anatomy. This is a nice picture from this paper of a tail. It shows the total length of the tail, how you measure that from the sacrum to the very tip of the tail. You see how the individual vertebrae vary in size along the length, going from fairly small to fairly small again at the very end, but in the middle you have the longest vertebra length, which ends up being predictive of the total tail length and also differences in tail size between forest and prairie ecotypes. Also important to realize they're studying this tail at a very young age in the embryo, well before it's been born. And these tails develop differently than roots. I'm a plant biologist, so I think of tails like roots, but they're really a different process of tail formation. These cells are differentiated very early in embryogenesis, and they differentiate from this presomictic mesoderm, the PSM cells. You see that dashed line between S-1 and S0. That's right at the border of the somite development. There would be thousands of cells in each of the somites, but there is a boundary that is created that then allows for, it leads to different vertebrae in the tail. The presomictic tissue is being controlled, in a way you could say, by the NMPs, the neuromesodermal progenitor cells, which are that pocket of cells at the very bottom. The very last slide, we're going to look into the comparison between the prairie and the forest mouse for the amount of presymictic tissue and the NMPs, and it turns out that that could be an important conclusion of how tail length varies. What I suggest is that the more cookie dough you have, the more PSM cookie dough you have, the more somites you can develop. Here is this model that they end up with, and there's another question about clock speed at the rate of partitioning these somites versus the amount of PSM tissue, and we'll get back to this at the end. All right, in the beginning, these mice are deer mice from the eastern US, natural populations that occur in Michigan, which apparently has a lot of prairie compared to eastern Pennsylvania, or sorry, western Pennsylvania or western New York, I forget which one, where the forest mice occur. There are different subspecies of the same species. They do interbreed, so there's gene flow, but these differences in tail length are maintained, which suggests that there's strong natural selection and also a genetic basis. And so this paper is really investigating what is the genetic basis for tail length variation. We know that these populations are recently diverged because the habitat is fairly recent too. 20 or 40,000 years ago, this was all under ice, or pretty much, I guess. At least the Michigan population would have been. But in the paper, they suggest that these populations have evolved in the last 2,000 years. It could have been that the prairie habitat is relatively new, and whereas the forest habitat is older, perhaps. We see this dramatic difference in tail length between the forest mice versus the prairie mice, much shorter tails. These are just two representatives of the forest and the tail, or sorry, the forest and the prairie. Difference in the habitat picture there too. Obviously lots of agriculture in Michigan as well. So this is a comparison between caudal vertebra, individual caudal vertebra going from 1 out to 25. For each one of those caudal vertebra, how long is it? This isn't the full tail length. The forest mice do have longer tails than prairie mice, but they also have longer individual caudal vertebrae. So long tails have more larger tail vertebrae. You can see that over on the right-hand side, the forest mice are the only ones that have caudal vertebra 26, 27. So they have more vertebra, and they also have larger tail vertebra. Every single one of those tail vertebrae, except the ones that are really close to the base of the tail, the sacrum. The earliest vertebrae are very similar in size, but then soon they diverge in size, and that leads to a longer tail in the forest mice. The longest tail vertebra is a good correlate of total tail length, and this is from F2 data, F2 populations that they created, but I thought it was useful just to show it right here. Long tails have larger tail vertebra. Oh wait, yeah, this is the longest tail vertebra on the x-axis and the total tail length on the y-axis. The number of tail vertebra is also strongly correlated with tail length. So both tail vertebra number and length of individual vertebra matter for total tail length. I love these data. It's a nice allometry presentation. This data is all from the F2 population, not from the parents, but I like the data. They do this bucket science test of whether performance of this rod crossing assay, I forget what they call it, but they're basically running the mice across this rod, and we saw the effects in those videos, and the forest mice excels at this, and the black bars that go down are the times that they fail. The prairie mice basically are like, I'm good, I'm going to stay on this side. Or if they do go, they fail. They just can't make it. I don't know if failing means falls off or if they turn back. But the forest mice are just like, any day, any time, I'll cross that rod. So there's obviously a difference in performance on walking across this rod, which is analogous to walking in trees, on branches, or small vegetation. So forest mice are better suited for that. All right, so F1 hybrids have intermediate tail lengths. And I got really excited at this point in the paper because I'm like, oh, what? Are they going to do a QTL study? And sure enough, they do. So they set up the F1 population, and then they breed the F1 and produce an F2 population. I put an exclamation point there beside the F2. Great job. They did an F2. And they have this variation in tail length in the F2 population. They've got the average of the prairie population, it's shorter, and the average of the forest population, it's longer. And then they have this distribution, a frequency distribution of the F2 individuals, which show quite a range of variation. Remember what Edward East says from our textbook, that only very rarely would the F2 population resemble the parental phenotypes, and that's certainly true in this case too. So it's conforming to Edward East's predictions. Edward East went on to do other things, but this is a great textbook tie-in. We've already learned about QTL, we've already learned about Edward East, and here's another recent example of the utility of QTL in discovering the genetic basis of a phenotype. This was, there's more data, different dimensions like the longest vertebra length in the F2 population also showed normal distribution, intermediate between the two parents, and didn't very frequently resemble the individual parents. So the next step in QTL is to find a series of SNPs, and since the deer mice are so closely related to lab mice, you can easily use the SNPs and single nucleotide polymorphisms, that is, between the prairie mice and the forest mice, to then be able to get closer to the genes that might be involved. And we've already seen this karyotype in a previous paper when in the sugar glider presentation they used a mouse model system, and I showed the karyotype from this paper. And some students pointed out during that, well, these are the actual chromosomes that end up holding the QTL, but I don't need to show that right now. But this one chromosome 8 in this picture happened to be a trisomy 8, and a student asked what's going on there. I didn't know at that point, and so I did some research, and there's actually been quite a bit of research about trisomy 8 in deer mice and mice in general. It seems to be a stable polymorphism where trisomy 8 is a mosaic. That is, in individuals, some cells will have a trisomy 8 and other cells won't. Assuming the gametes don't have the trisomy 8, otherwise they probably wouldn't be successful, whereas some cells do have this trisomy 8. The adaptive value of that, who knows, or is it just a mutation that happens here in deer mice? I don't know. But it is interesting to note that Hox D13, the D Hox cassette, is on chromosome 8. This paper never talks about the trisomy 8. So if Hopi and Evan were here, I'd ask them, so were your mice trisomy 8? Did you check? I don't know. Is there something going on there? I don't know. But something to consider. This is, I consider it a fun fact. It's not really related to this paper that much. Deer mice in general have 24 chromosomes. 2 of them are behind my image. There's the 23rd and then the sex chromosomes. So it seems like at least one of the parental species doesn't seem to have it. From an old paper in the Journal of Mammalogy, I found a karyotype of the Maniculatus nubitree. Didn't have it, wasn't showed here, but of course in a karyotype you can just pick and choose the chromosomes and just ignore the cells that had 3, and who knows, maybe they just didn't see it, but something to consider. All right. For their QTL study, they showed graphs like this. The QTL is not shown yet, but it does show the SNPs on the x-axis and the chromosomes that ended up being important in the QTL analysis. It's not showing all 24 chromosomes, just the ones that ended up having the QTL on them. But the little hatch marks on the x-axis are the individual SNPs that were polymorphic between the forest and the prairie ecotypes. So that's cool. I tell my students you're going to have to find lots of SNPs spread throughout the entire genome, and there you go. They found them. So this is the QTL map for total tail length, and the QTL is measured in the LOD score, logarithm of the odds, which I call the QTL score, just for simplicity. The line that goes up and down, once it crosses that dashed line, is essentially the level of the significance threshold, and then you form this window around that up part which is kind of the region of the genome. If you intersect it all the way down on the x-axis, that's the region of the genome where you'd expect the gene or genes that control tail length to reside. So this is narrowing down the scope of the entire genome to just a smaller area of the genome to look for genes, and we'll see how they get to HOXD13 in a lot of this presentation. So these QTLs that I'm showing here, those gray bars, explain 24% of the variation in tail length, which is actually pretty good for a QTL analysis. Any multivariate test, 24% explanation is pretty good. And I just like to say that for my students who might be used to seeing a much higher percentage of variation explained. 24% is pretty good on a polygenic trait like tail length, and I just threw that out, that HOXD13 is one of the many genes that may be involved, but this paper kind of highlights the effect of HOXD13. Here's another QTL map of the longest tail vertebra. It's on 3 of the 6 QTLs that were identified for tail length, and then the number of tail vertebrae are on the other 3 of the 6. So in the end, we're going to study the number of tail vertebrae as being important. And it wasn't quite clear to me why they focused mostly on the number of tail vertebrae, but they did, and that's where they really isolated HOXD13. So within this now, we can identify candidate genes in the range, or actually just the number of genes, just genes. What genes are there? Count them up. And when you do that, there's 527 genes in that region of chromosome 3. There's 25 genes in that region of chromosome 8. And there's 110 genes on chromosome 14 in that region. I took a deep breath because that's less than the 20,000 genes, but now we've isolated it down to about, let's say, 700 genes that we have to consider one by one, kind of, in a way. Next thing they do is really logical, and it's cool that in genetics we're getting to a point where it's like, well, just compare the forest versus the prairie, see if those genes are differentially expressed. Seems simple, right? I'm laughing because it didn't used to be simple, and I'm sure it wasn't simple, but now with RNA sequencing you can actually do that, compare the forest to the prairie embryos, and look at that tissue, just excise that tissue in the red circle and compare the genes that are expressed early in development in the PSM, ideally, or at least in the embryonic tail bud. Look at the genes that are expressed at this early embryonic stage, 12.5 days. E12.5 stands for 12.5 days old versus later, 15.5 days old. You could compare between early and late, and they'll do that too, but really we're looking for this comparison between a change in expression from forest to prairie. That change would be a gene that we would be interested in, especially if it falls in one of those QTLs. So at E12.5 days, that is embryonic 12.5 days old, forest versus prairie, is there a change in expression? If there is, that makes that gene a stronger candidate for being a critical gene for tail development. You could compare it at late embryo too, at 15.5 days, change in expression, and they did that, and that's the only reason I'm showing this one here too. When there was a differential expression early and late in the same direction, then that helped them suggest that, yeah, this is probably an important gene. So when they did this RNA-seq, you have these 527 genes in chromosome 3. Which ones are differentially expressed? 112 are differentially expressed. What about chromosome 8? 85 to 28. 28 genes are differentially expressed. What about chromosome 14? Goes from 110 genes down to 28 genes also. CG stands for candidate genes. So they've narrowed down the set of candidate genes to about closer to 150 genes. But then how do you get to HOXD13? Still, this is the question. Take these 150 genes and then basically look into this mouse genome informatics database, which is literally mousegenome.com. Type it into the search menu right there, and each gene, one by one, you can do a search term like, does it have tail length? Does it have vertebrae on it? Does it have size? Anything like that. And when they do that, it comes back with 5 candidate genes: SP5, HOXD13, HOXD9, HOXA10, and APC. Those 5 genes had, in this gene orthology database, some effect on tail vertebra size. I told my students this kind of seems a little bit circular, like why didn't you just look it up on the mouse genome informatics database? Well, you could have done that, but you would have had no confirmatory research that suggested that those genes were differentially expressed, like the RNA-seq study that they did, or that in a QTL turned out to be in a QTL region in the genome. They had both of those and then went to the gene orthology database. So that's really strong. They're getting closer. Next thing they're going to do is look at allele-specific effects in the F1 population. So they look at each one of these genes. We'll start with HOXA10, looking at the forest, which is going to be a green dot, versus the prairie allele, which is going to be the tan dot. In the F1 hybrids, the RNA-seq counts in early and late. This is what the data look like. The forest RNA-seq count for HOXA10 versus the prairie HOXA10 expression. Not significantly different. So there's going to be a little bracket with a star if it is significantly different. In this case, it was not in early. It was also not in late. But you can see that there is a change in expression early to late. It's kind of confirmatory. Genes expression is changing through time, through embryonic development. That is cool to see. It's really cool to see, but it didn't tell a story that was concomitant with tail length variation. So we're going to, fortunately, in a way, be able to rule out HOXA10. And then we go on to SP5, and we see there is a significant difference between early prairie versus forest RNA or expression of that gene. However, it goes in the opposite direction it did from the parents, like the prairie versus the forest. So they're a little bit concerned that this might have been a epigenetic effect. As we'll see, they kind of are unsure whether SP5 should be included, and they kind of rule it out. Clearly from the title of the paper, they didn't have SP5 in the title. Again, that was because it went in the opposite direction as what they'd seen between the parents of the F1. But then HOXD13, early and late, significant differences, significant decline in expression. So there's less expression of HOXD13. Maybe that's what's leading to continued pre-mesodermal cell proliferation or continued stem cell population growth, that is more stem cells producing more cookie dough for tail formation. So they're pretty happy about that result, I would say. HOXD13 seems to be the major player, and then the other 2 genes don't turn out to have an allele-specific effect. So this is like getting way down into the weeds, but that is HOXD13, and we're left wondering with the next step, well, what about choosing fine scale mapping? HOXD13 is sitting right there on chromosome 8. But if we get some SNPs that are even closer to HOXD13, that might confirm that that region of DNA is actually important. The dashed vertical lines, not the ones that are going out, but the vertical lines are these fine scale mapping SNPs that they found. And if we do the allele specific test on those SNPs, we see actually that the forest homozygote has more or a greater number of caudal vertebrae, and it's significant. You see the P03, P.03 at the bottom. So the forest allele and the forest homozygote for those SNPs have a greater number of caudal vertebrae, and that's the value of having this whole F2 population. You can do this fine scale mapping and then just go back to that data and see how many caudal vertebrae they had, and it's an incredible extension, confirmatory, really narrowing it down to HOXD13. That was for the left-hand SNP. The right-hand SNP also shows a greater number of caudal vertebrae for the forest homozygote of that SNP. It's literally of that SNP, forest homozygote, more caudal vertebrae. Note that SP5 and HOXD9 are close by on chromosome 8, but that's not what they're studying in the graph above. What they're studying is the SNPs. In a way, I could have just blocked out SP5 and HOXD9. I probably should have, just to make it more clear. Then they asked the question, is it the HOXD13 protein? Notice I wrote that in all caps. All capital letters refers to the protein. They show the prairie versus the forest that there is a 3 base pair insertion in the prairie. Now I'm biasing it. I'm saying there's a 3 base pair insertion. We could have said it's a 3 base pair deletion in the forest one, but what we can tell looking across other animals is that most other mammals don't have this insertion. So it's only the prairie deer mouse that has this extra 3 base pairs, which leads to another alanine, which leads to a polyalanine track. There's 5 alanines in a row. This is a polyalination, I guess you could say. And so there is a difference in the protein between the prairie and the forest ecotype. So these days, what's the phenotypic effect of that? It turns out, just by kind of analogy, in humans, polyalanine tracks in HOXD13 are known to cause syn polydactyly, the fused digits, so it has some effect, this polyalination, on bone formation or digit formation. The bones, maybe not. I'd have to take a closer look, but yeah, it seems like the bones are kind of messed up there too. But these days, if you're asking about the protein or you're getting closer, it seems, I'm sure the editor's like, well, you should really do the CRISPR test. And so they're like, okay. And they went and did the CRISPR test. They used CRISPR-Cas9 to introduce a polyalanine track in HOXD13 in lab mice. They just basically inserted those extra 3 base pairs in lab mice and then looked at the tail of the CRISPR versus non-CRISPR, and nothing happened. They were probably, well, it just is what it is in science. You just take the results, report them, and there was no significant result in the CRISPR test. But they crossed that bar. I'm sure there's a story of like whether they thought of doing this on their own or if the editors required it. Who knows? I'd love to know more. But kudos for reporting this negative result. I always try to emphasize to my students how important it is to publish negative results, and this is a great example of where the CRISPR didn't work out. We see so many papers where the CRISPR works out perfectly, but I know it's not always the case. Those of you who've done CRISPR know that it's a mosaic chromosome situation too. Some mice have it, some mice don't. It's difficult. So is it the HOXD13 protein? No, or maybe not. I'm using the qualifier there. We're not quite sure, but it's probably having to do with changes in regulation. I'll just go back to Sean Carroll's Endless Forms Most Beautiful, a direct quote from the book that I think is useful here. And the authors of the study would appreciate this because the first 2 citations in their paper are from Sean B. Carroll. Diversity is not so much a matter of the complement of genes in an animal's toolkit, but in the words of Eric Clapton, it's in the way that you use it. The development of form depends on the turning on and off of genes at different times and places in the course of development. And it appears that this may be what is happening with Hox D13. So a last glossary note slide, and then we'll have one last figure before we're done. Syn polydactyly is just fused digits. Polyalanine tracks are a streak of alanines in a protein. RNA-seq is RNA sequencing. Mosaic chromosome sets are when some cells in an organism have a different number or kind of chromosomes. Trisomy 8, and that's common in deer mice as mosaic chromosomes, and it didn't seem to affect these mice in their study. But lastly, this is their final model of how tail length varies, and if you remember the presymictic tissue, which was below the last horizontal line going down on the tail, between the last horizontal line and that population of little dots at the bottom, those are the NMPs, the neuromesodermal progenitor cells. The area that has no horizontal lines, that's the presymictic tissue. And you see in the forest mouse, there's more presymictic tissue, and there's also more neuromesodermal progenitor cells. So perhaps this decrease in Hox D13 expression in forest mice leads to more NMPs and more presymictic tissue, and that's what leads to more longer vertebrae that leads to a longer tail. And what I say is that if you have more cookie dough, you can make more cookies, and maybe that's what's happening here in the deer mice. I always go back to my take-home points. Tail length variation has recently evolved between prairie and forest deer mice in the eastern U.S. Tail length has a performance advantage from those fun tests that they showed, and QTL analysis, RNA-seq, and gene phenotype public information all point to the Hox D13 being a gene with a major effect on tail length. There's certainly other genes involved. CRISPR did not confirm. Kudos again for reporting those negative results. And then it must be, perhaps, qualifying again, cis-regulatory effects on Hox D13 and other genes during embryogenesis that control gene tail length. I was a little bit disappointed. I'll just say that in the title of the paper, it ends up having Hox D13. Only 24%, that QTL analysis shows 24% of explaining tail length variation. There's certainly other genes, and this TBXT gene is that involved too? It's interesting. Why it would have been interesting to check for TBXT in the RNA-seq analysis. Maybe TBXT has an effect earlier in embryogenesis at like day 0, day 1, day 2. And this window of when to look, that must have been tearing your hair out, like when to actually test. But kudos, Hopi and Evan and all the other investigators on this paper. It was a pleasure to read. The paper was incredibly well written and very easy to understand, and I really appreciate that. And Hopi, I hope you continue to produce papers like this for us to all learn from. All right, bye-bye.